Alright, so today we're diving into the backbone of life itself, polypeptides and the peptide bonds that link amino acids to form these essential chains. Understanding the peptide bond is crucial because it's the foundation of protein structure, and as we know, proteins do pretty much everything in biological systems. Let's start with the basics. Peptide bonds form through a process called dehydration synthesis, also known as a condensation reaction. In this reaction, the carboxyl group of one amino acid reacts with the amino group of another. This releases a water molecule and forms a new amide linkage, which we call a peptide bond. For example, when glycine and alanine link together, glycine's carboxyl group bonds with alanine's amino group, releasing water in the process. This forms a dipeptide with a specific direction. A free amino group on one end, called the amino terminus or N-terminus, and a free carboxyl group on the other, called the carboxyl terminus or C-terminus. Now, as more amino acids join together, we get a polypeptide chain. The backbone of this chain consists of repeating nitrogen and carbon atoms. Each segment of the backbone corresponds to an amino acid residue, and this regular pattern is what gives polypeptides their stability and structure. Polypeptides are always synthesized in one direction, starting at the N-terminus and ending at the C-terminus. This reflects the order in which amino acids are added during protein synthesis, with the ribosome playing the role of the assembler. But here's the catch. Peptide bond formation isn't thermodynamically favorable. In fact, it requires an input of energy. As we can see from this free energy diagram, the transition state has a higher energy than the individual amino acids, which means the reaction needs an energy push to proceed. So how does the cell get around this energy barrier? It uses ATP and GTP, which are molecules packed with energy. These energy-rich molecules power the ribosome, the cellular machine responsible for linking amino acids together during protein synthesis. Once these peptide bonds form, they are quite stable. However, proteins don't last forever. Cells regulate their breakdown through a process called proteolysis. Proteolytic enzymes, or proteases, cleave peptide bonds at specific sites within the protein. For example, trypsin cleaves at the carboxyl side of lysine and arginine, while chymotrypsin targets hydrophobic amino acids like phenylalanine and tyrosine. Now that we understand the basics of peptide bond formation and cleavage, it's important to recognize how this impacts the final structure of proteins. The polypeptide chain folds into complex shapes, alpha helices, beta sheets, thanks to interactions between the backbone and side chains, ultimately determining the protein's function. Interestingly, while forming peptide bonds requires energy, breaking them down through hydrolysis is thermodynamically favorable. However, this process is kinetically slow, meaning it doesn't happen quickly under normal conditions, which is why proteins are stable in cells. So, to wrap things up, peptide bonds are the key to building polypeptides, which in turn form the backbone of proteins. From synthesis powered by ATP to breakdown via proteases, the formation and cleavage of these bonds are crucial to life at the molecular level. If you're looking to dive deeper into topics like these or want to test your understanding, check out our King of the Curve app. It's your go-to resource for mastering topics like this and so much more. Download it now or visit kingofthecurve.org to get started.